Hello everyone. Today we'll be solving Cambridge International AS and A Level Biology Paper 5 Planning, Analysis and Evaluation. February-March 2021 Paper 52. Question number 1A. Students at a college often took a shortcut between the library and the tennis court across an area of open land. So we can see the tennis court and we can see the library, and there is this shortcut that the student often took. All right. The area of open land was covered by a large number of plants of a variety of different species. Walking over this area many times damaged the plants by trampling. As a result, a shortcut path formed across the area of open land between the library and tennis courts. This path was approximately 1 meter wide. Figure 1.1 shows a map of the area. Okay, so we can see the map of the area. Footpath that is linking the library to the tennis court. However, people love to go through the shortcut. Some student decided to investigate the effect of trampling on the biodiversity of plant species growing in the area of open land. State the data the student need to collect to assess the biodiversity of the plant species. In order to calculate the biodiversity of the plant species, we need to know the number of different species of plant and the abundance of each species of plant that is available in that particular place. Part 2. State the independent variable in this investigation. So the independent variable is whether the area got trampled versus the area did not get trampled. So trampled versus non-trampled. Part B. Describe a method that the students could use to compare the biodiversity of plant species growing in the area of untrampled open land with the biodiversity of plant species growing on the shortcut path. Your method should be set out in a logical order and be detailed enough for another person to follow it. So, answer like this, first of all, we need to mention what size of quadrant we are going to be using and what kind of transect, the mapping system we're going to be using. So we can use a 0.25 meter by 0.25 meter quadrant because that particular place is a relatively small space, and we can use a belt interrupted transect. The trampled and untrampled areas should be sampled. The quadrants then can be placed like at an interval, example like 2 meter along the transect, wherever the transect are basically drawn in that particular piece of land. The species of plant can be identified using a catalog of known species found in that area, and the number of individual species was counted, and care should be taken to note low growing, easy to miss species, those that can be present in that particular place. At least 3 separate belt transect in the untrampled and trampled areas should be recorded. The whole investigation should be repeated at different times of the year, and Simpson's index of biodiversity should be calculated for fair comparisons in different times of the year, because different times of the year will have different amount of plants, different kinds of plants present over there. Some plants may cause allergy, so the person who is conducting the experiment, should wear protective clothing and should wear gloves to prevent allergy. Question C. The student compared the drainage of water through the soil from the shortcut path and with the drainage of water through the soil from the untrampled area of open land. The student obtained soil samples by pushing the tube-shaped cutters vertically down into the soil to a depth of 10 cm. Each tube-shaped cutter had a diameter of 10 centimeter. When removed, a cylinder of soil 10 centimeter in length remained in each cutter. Figure 1.2 shows one of the tube-shaped cutters. Alright, we can see the cutting edge. Alright, and 10 centimeter of a block and 10 centimeter diameter was taken. Soil samples were collected at random from the shortcut path and from the untrampled area of open land. The student collected 10 soil samples from each of the two areas. Drainage was measured by pouring the same volume of water into the top of each of the tubes containing soil. The time taken for 10 cm3 of water to drain through each tube was then recorded. Okay. The table 1.1 shows the results that the students recorded. The shortcut path time needed for 10 cm3 of water to drain through the soil is always higher than that of the untrampled area. As we can see, 125, 148, 192. All right, and then there is mean and standard deviation has been done. So in untrampled area, the water drained faster. Okay. From the results shown in table 1.1, the students concluded that trampling compresses the soil, reducing the size of air spaces. This means that water drains through the soil more slowly. State one way that the results in table 1.1 support the conclusion and one way that the results in table 1.1 does not support the conclusion. So in favor, all the time in trampled area, water drains to the soil more slowly than untrampled area. So we can see in every one of them, it is slower in every one of them. So that's why it supports the data. All right, it supports the conclusion. Now, why it doesn't support the conclusion is that there is no information about soil airspace. There is no information about soil airspace given whether in the trampled or untrampled area. So that's why it does not support. The student calculated the standard error SM and 95% confidence intervals. 95% confidence intervals for the drainage data shown in table 1.1. The formula that student used were 95% confidence intervals, mean average plus minus 2 into standard error. Okay. So S stands for standard deviation, N is the sample size, the number of observation, and table 1.2 shows the calculated values of SM and 95% confidence interval for the data from the shortcut path. We can see the standard error SM 9.6, 95% confidence intervals 137.1 to 175.5. All right. Now the question says complete the table 1.2 to show the calculated values for SM and 95% confidence interval for the data from the untrampled area of open land. So to find out the standard error, the formula that is given here, standard error is equals to S divided by standard error for untrampled. Standard error for untrampled is equals to, all right, standard deviation, untrampled area standard deviation is 9.0 divided by square root of 10, which is the number of terms, equals to 2.8. Now the 95% confidence interval is equals to 68.2 to 79.4. The way we come to this particular conclusion is basically we do the plus minus. So here we had 73.8, so the method should be 73.8 plus 2 into 2.8. So that is equals to 79.4. and for the lowest value it is going to be 73.8 minus 2 into 2.8, which is equals to 68.2. So that's how we get the confidence, 95% confidence interval. Calculating 95% confidence interval provides additional information for interpreting the results of the investigation. State the additional information about the data shown in table 1.1 that can be gained from the calculated values of 95% confidence interval. So basically there is a 95% probability that the true average lies within these limits of 68.2 to 79.4 in the case of this untrampled area. So the difference between the average is likely to be significant at probability less than 0.5 because there is no overlap between 95% confidence interval for untrampled and trampled area. The narrower the confidence interval in the untrampled area, the closer the calculated average is to the original average. So that's how basically we can use this information. Part D. In a further investigation, the students studied one species of plant growing in the area of open land. They noticed that some plants of this species had larger leaves than other plants of this species. They thought that the surface area of the leaves might be affected by the drainage of the soil. The student collected 20 plants of this species at random. At each place where a plant had been collected, the student collected a 10 cm deep soil sample using the tube-shaped cutter as shown in figure 1.2. For each plant, the student determined the surface area of the largest leaf. Outline how the surface area of the leaf can be determined. So to get the surface area of a leaf, we can place the leaf on a grid, basically on a graph paper or on a grid, and then outline the leaf, all right, like a trace. Then the square, those that are fully covered, and the square, those that are more than half covered, should be counted, and that will give us a very good approximate area of that particular leaf. For each soil sample, the student measured the time taken for 10 cm³ of water to drain through the soil sample as described in C. The students calculated the spearman's rank correlation coefficient for their data on leaf surface area and the time taken for 10 cm³ of water to drain through the soil sample. The students calculated the RS value as negative 0.455. State the relationship between leaf surface area and the time taken for the 10 cm³ of water to drain through the soil sample. That is suggested by an RS value that is less than 0. So basically from this particular value we can see that there is a negative correlation because there is a minus sign in front. Okay, as the time taken for 10 cm³ of water to drain decreases when the leaf surface area increases. Table 1.3 shows part of a table showing critical values of RS at different probabilities P. These critical values are used for testing the null hypothesis that there is no correlation between two sets of data. Our exam probability is 0.05 and the number of pairs of terms in the sample in this case that we are doing is actually 20, so it will be 0.447. To use the table 1.3, negative RS value must first be converted to positive values. Using the positive RS values of 0.455, state and explain what can be concluded from the table 1.3 about the relationship between leaf surface area and the time taken for the 10 cm³ of water to drain through the soil sample. So the working, all right, will be at 0.05 as we have already shown. The relationship is significant and not due to chance. The relationship So the relationship is significant and not due to chance, as calculated RS 0.455 is greater than the critical value of 0.447. So basically, whenever the probability at 0.05 and the number of pairs of items in the sample is equals to 20, it gives us 0.447. Any value that is greater than that, we are going to reject the null hypothesis, as the calculated RS 0.455 is greater than the critical value of 0.447. Question number 2a. Indigo carmine is a water-soluble indicator. When oxygen is bubbled through a solution of this indicator, the color changes from colorless to blue. A student used indigo carmine solution to investigate some of the factors that may limit the rate of photosynthesis. Figure 2.1 shows the apparatus used by the student. 15 cm cube of colorless indigo carmine solution and a 5 cm cube of sodium hydrogen carbonate solution were placed. Large test tube, alginate beads containing immobilized single-celled aquatic photosynthetic protocysts were also placed. The student prepared 4 sets of this apparatus. For each set of apparatus, the student used a different concentration of 5 cm cube sodium hydrogen carbonate solution, 0.25, 0.5, 0.75 and 1.0. Sodium hydrogen carbonate solution is a source of carbon dioxide for this photosynthesis process. After adding indigo carmine solution and sodium hydrogen carbonate solution to the alginate beads, the apparatus was illuminated with a bench lamp from a distance of 20 cm. To minimize heating effects, the bench lamp used an LED bulb. For each concentration of sodium hydrogen carbonate solution, the time for the indigo carmine solution to turn from colorless to blue was recorded. The investigation was repeated for 4 new sets of apparatus illuminated with the same bench lamp at the distance of 10 cm instead of 20 cm. The same concentration of sodium hydrogen carbonate solution were used as previously. All other variables in this investigation were standardized. State the dependent variable in this investigation. So basically, you see, we are testing with a dye, which is indigo carmine. All right, and basically what happens is that it is a water soluble indicator. When oxygen is bubbled through a solution of this, the color changes from colorless to blue, so the dye will become blue. So what we need to do is basically we need to measure the time taken for the indigo carmine solution to turn blue. Part 2. State one variable that needs to be standardized in this investigation other than the temperature or those variables clearly standardized in figure 2.1. So you see, the mass of the beads of immobilized enzyme should also be standardized. Other factors that can be kept constant is pH, concentration of the indicator, and concentration of alginate. Part B. The time taken for each indigo carmine solution to turn blue was converted to a rate of photosynthesis in arbitrary units. This was done in such a way that the slowest rate of photosynthesis in this investigation was given a value of 1 arbitrary unit, and all other rate of photosynthesis were therefore greater than 1 arbitrary unit. Table 2.1 shows the results of this investigation and some of the students' calculated values. Distance of apparatus from the bench lamp, 20 centimeter. So we can see percentage concentration of sodium hydrogen carbonate 0.25. The time taken for the indicator to turn blue is 600, and the rate of photosynthesis in arbitrary unit is 1.0. So definitely with this understanding, With this understanding, we can understand that whenever we are finding for 1.94, what we are doing is we are going to divide this 600 by 310. So that gives us the result 1.94. So for the 10, for this experiment with 235 seconds, so it's going to be 600 divided by 235, which will give us a result of 2.55. Complete the table by calculating the missing rate of photosynthesis. We already did. State 3 conclusions that can be made from the results shown in table 2.1. The factors that affect the rate of photosynthesis and when these factors become limiting factor. So we can see in all of this, increasing the sodium hydrogen carbonate concentration increases the rate of reaction. However, it is only limiting until 0.75 mole per dm cube sodium hydrogen carbonate solution. All right, in both experiment. All right, we can see, but in the second experiment, we can see that increasing the sodium hydrogen carbonate solution keeps on increasing the rate of photosynthesis. So increasing hydrogen carbonate concentration increases rate of photosynthesis. Also, we can understand increasing light intensity increases rate of photosynthesis. So the rate of photosynthesis increases the rate of photosynthesis. So we can conclude that increasing hydrogen carbonate concentration increases the rate of photosynthesis. We can see it from here. Once we increase the hydrogen carbonate concentration, we increase the rate of photosynthesis. We can see the rate of photosynthesis in arbitrary unit increases. Increasing the light intensity increases the rate of photosynthesis. We can see with the same hydrogen carbonate, with the same hydrogen carbonate concentration, with the same hydrogen carbonate concentration, we can get the time taken for the indicator to change color 195, which indicates that it takes a slower amount of time and thereby faster rate of photosynthesis. And at 0.75 sodium hydrogen carbonate and the distance of 20 centimeter, we can see that light intensity becomes limiting factor because the rate of photosynthesis is no longer increasing. At this point, CO2 concentration was no longer the limiting factor. And finally, at 10% sodium hydrogen carbonate, all right, we can see that the at 1% sodium hydrogen carbonate, we can actually see that increasing carbon dioxide concentration is still keeping on increasing the rate of photosynthesis. That means light intensity is no longer limiting in this particular. Part 3. Confidence in conclusions that are made from the results shown in table 2.1 is reduced by a lack of repeats and lack of statistical analysis. Suggest 2 other reasons that reduces confidence in conclusions that are made from the results shown in table 2.1. So the question says, suggest two other reasons that reduces confidence in conclusions that are made from the results shown in table 2.1. So let's look into the conclusions. So the conclusions were increasing hydrogen carbonate concentration increases the rate of photosynthesis. All right. Increasing light intensity increases rate of photosynthesis. We can prove that because only a small range of different light intensities were used in the experiment. Light intensity at 10 centimeter or at 20 centimeter might just be the same, so it is not a fair comparison. And the color change that is actually within the experiment is actually subjective, and it is difficult to judge when the color change has actually occurred. And we can see that this experiment is only done for one species of Protoctist only, so the results may not be valid for all other species. Guys, we are done with the question paper today. Thank you for joining the video. If you like the video, then like, subscribe the channel, and if you want videos like this to be posted more, then write us in the comments what videos that you want us to solve next. Thank you for joining the video today. Bye bye.